Am I audible?
Yes, you're audible.
So can you hear me? So, hello everyone. Welcome to Progress and Prospects in Biology, the Sunday webinar series. So we are back with today's talk. Today, we have with us someone very special, Professor Shantanu Ray, uh, Systems Ecology and Ecological Modeling Laboratory, Department of Zoology, Vishwabharati, Shantiniketan. Before we begin today's talk, there are few things which I would like to tell our viewers. We have a new website. Please check out our new website. It is ppbiology.wordpress.com, and we will have lots of new contents coming up. Please check out our website. We are going to have a conference coming up, which is called The Half of It Women of the Bio World. The poster is up on our website. Please check out the poster. Please also check out our YouTube channel, our archive talks, and please use the holiday season to look at the content we have done before. So, thank you for your continued support. Thank you, all the viewers who are here with us today. And with that, I'll turn it over to our mentor, Professor N.R.A. Banerjee, who's going to give the welcome address and briefly introduce today's speaker. Over to you, ma'am.
Thank you, Rupatha, and thank you, Shia. A very warm welcome to all our viewers tonight, and a special welcome to our speaker tonight, Professor Shantanu Ray, as Rupatha said, and I have the honor to introduce him. So before that, I must mention that both of us shared the same genealogy, right, Shantanu?
Yes,
The department as also the lab. But
yes.
Shantanuda has specialized in something that students of biological sciences are usually quite afraid of. So today we are going to learn something very, very novel, and the concept and the ramifications thereof of such a concept, and the potential to extrapolate it virtually to every domain of the sciences is something that we shall learn from Professor Rai tonight. So, progress and prospects in biology, as you all know, we are running continuously for 6 months. And every weekend, we try to bring to you by our Sunday webinars the latest in the world of biosciences. And on Saturdays, we bring to you special lectures from the world of biology as well as non-biology. And last Saturday, we were honored to host Professor Bernard Patton, whose direct student is our speaker tonight. So I introduce you, Shantanuda, with a very brief introduction. It does not do justice to the enormous body of work that you have done. So here goes. Our speaker, Professor Santanu Ray, is currently Professor, Systems Ecology and Ecological Modeling Laboratory, Department of Zoology, and he's also the Director and Advisor of Indira Gandhi Center and Foreign Students Assistance Cell of the Vishwabharati University, Shantiniketan. His areas of research are systems ecology and integrated ecosystem theories and their application, ecological phenomenology and ecological modeling, static, dynamic, structural dynamic, and theoretical modeling, statistical modeling using neural network on mangrove, estuarine, and reservoir systems. He has more than 140 publications in refereed journals and international book chapters in books. His postdoctoral work experience and teaching experience have been with legends like Milan Strasskraba, Czech Academy of Science, Sven Erik Jorgensen, University of Copenhagen, Eugene P. Odom, and Bernard Christopher Patton of Odom School of Ecology, University of Georgia. And like I mentioned, we were honored to have hosted Professor Patton, and we hope he shall come back. And tonight, we shall learn from Professor Rai, who is also the associate editor of the following journals. Ecological Modeling, Energy, Ecology and Environment by Springer. He is also the section editor of Ecosystem Encyclopedia of Ecology by Oxford Elsevier, and he is on the editorial board of journals such as Ecological Informatics, Network Biology, Computational Ecology and Software, Journal of Ecosystem, Chinese Journal of Population Resources and Environment. He is also the secretary of Australasia Chapter International Society for Ecological Modeling, and he is the nodal member of National Network of Mathematical and Computational Biology in India. And while I was reading the CV, Shantanuda, I'm hoping that we shall learn many of the concepts that I read without understanding much. So a very warm welcome to you, Professor Rai, and now it's over to you.
Thank you, Anna. You are like my sister, and whatever you told about me, maybe I do not deserve all these things, but anyway, it is thankful to you. I am thankful to you for your kind introduction. So today, my lecture is different perspectives of systems ecology with case studies. Actually, I am telling you, my lecture is a little bit broad today, not a very pinpointed lecture for a particular subject. If I penetrate vertically of a particular aspect, that will be very, very difficult for the general audience, because I know in this particular lecture series, there are many people, they are actually trained in parasitology, genetics, ecology, or whatnot, many other fields. But it should not be very specific for a modeler. That's why today I have chosen the topic. I selected the topic, which is little bit broad, but I will go little bit in deep. And obviously, whatever I will give you the case studies which was done in my lab. Okay. Now I'm coming to the PowerPoint. And one thing I'm telling you, I'm thankful to all the organizers for allowing me to deliver the lecture in this platform, because whatever I've seen the name, the past, speakers. So I'm really delighted to be actually approved by the organizers as a speaker. Okay. Now I'm coming to the share screen. Oh, can you the slide is visible to all of you? Hello. Slide is visible?
Yes, sir, it's visible, sir.
Okay. Now I can start. Okay.
Yes, sir, the slide is visible. Please.
Okay. So the title of the talk is Different Perspective of Systems Ecology with Case Studies. And I will show you my case studies of my lab works. And if you need the paper, if you will go to the website, which is written below, in the website, if you click, then most of the papers you can get it from, or rather you can ask me through email. I can send you the papers if you like. Okay. So the first I'm telling about what is systems ecology. Actually, you know about ecology. Actually, you have learned ecology from your school life. And what is systems ecology? Whenever you started biology, you know what is biology and what is the cell. Cell is the structure and functional unit of the body. And different cells combine together to form the tissues. Different tissues combine together to form the organ, and different organ combines together to form the systems, and different system constitute a body or the individual. Now the ecology starts from here. Suppose the individual. So there are many species. So every species having many individuals. So all these individuals of a particular species constitute the population of that particular species. Am I right? And now there are different population of A species, population of B species, C species, and so on. So different populations considered and combined and ultimately form the community. So there are many communities and different communities combined together to form a system or ecosystem, ecological system. So whenever the ecological study is confined only to population, it is called population ecology. Whenever it is confined to community, then it is called the community ecology. Whenever it is now little bit extended to systems, whole ecosystem, then it is called the ecosystem ecology or the systems ecology. First of all, one thing I am telling you, so the ecological system is a bounded system or it is a uh, closed system. Is a closed system or is an open system rather. So I can say like that, for the simplicity of the study, we always think that the system is closed, but system is never closed. You cannot identify this is the particular system which is closed, which is not connected to the other system. So if you think in a broader way, none of the system is closed. Every system is open. So in that case, we call it the systems ecology particular. Whenever you confine to a system, particular system, then we call the systems ecology. Now the question is the whole system or the part of the system? So there are different modeling techniques regarding the systems ecology. I will tell about the whole, rather the different techniques of the. Now I'm coming to the systems ecology first. Systems ecology is an interdisciplinary field of ecology that takes a holistic approach to the study of ecological system. And obviously, if you consider the holistic approach, then you have to consider all the components of the system, major component of the system. It's starting from unicellular organism to higher mammals. So obviously all plants and all animals, and if you consider all these things in your study, it will be very complicated. So in that case, systems ecology, you cannot handle the systems ecology without the knowledge of mathematics. So without the application of mathematics, systems ecology is not at all possible. So central to the systems ecology approach is the idea that an ecosystem is a complex system, obviously complex. Any biological system is a complex. Suppose in a human being, one individual, if you think about the complexity of the human being, the organs, molecular level is a very complex. And think about 100,000 individuals in a systems, their interactions. So obviously the ecosystem is more complex, and I think is the most complex system of the world. Any man-made system is much more less complex than that of the ecosystem, even in an individual. So it focuses on interactions and transactions within and between biological and ecological system that is specially concerned with the way the functioning of ecosystem can be influenced by human intervention. You know, without energy, nothing is possible. Even single simple smile cannot be possible without energy. And where the energy is coming from? Actually, the energy is coming from the sun, the solar energy. That's why in every religion, almost in every religion, sun is considered as a god, because whatever the energy we have, whatever. Now I'm just actually utilizing energy to deliver the talk. You are seeing you are seeing me or seeing my lecture or slides, so that needs also energy. All the energy came from the sun. So how it came? Not directly, it came through food chain. Suppose the old process, photosynthesis, is the only process by which the solar kinetic energy is transformed into potential energy, and then that energy is transferred through food chain ultimately to the higher trophic level. So it's a very complex system. So systems, and that's why ecological system is always related with the thermodynamics. The first and second law, energy is neither created nor destroyed. And the second law is whenever one form of energy is transformed into any other form of energy, but during the transformation, some part of the energy is not lost, some part of the energy is dissipated or entropy. Okay. So in that way, the system is going on. And systems ecology uses the extends concepts from thermodynamics. That's why I'm telling thermodynamics. Now I'm telling about the person. those who are actually the thinker, great contributor of the systems ecology. So this is the picture of Howard Thomas Odum. You know the person, particularly Odum. Odum means there are two Odums, two brothers. Eugene P. Odum is the elder brother of Howard Odum, is 11 years elder, and both have died during 2002, 10th August, Eugene, and 20th August, Howard Odum. And Howard Thomas Odum, popularly known as Tom or just HT, is considered as father of systems ecology. And uh, the last week, whenever my teacher, Professor Barnard Patton, told Howard Odum he is actually hero of the ecological science, particularly the systems ecology, and he is the actually pioneer person in systems ecology. That's why he is actually known as the father of systems ecology. And he is in the fraternity, ecological fraternity, he is also known as HT or Tom. Okay. So he, along with his brother Eugene P. Odum, during 1967 proposed the emerging field of systems ecology. And nowadays, systems biology is very, very prominent, or the very important field in the biological science. In 1972, Eugene wrote a book on ecology. Actually, most of you have read this book, have gone through this book during your student life, that is Fundamentals of Ecology. And in that book, there was a chapter, Systems Ecology, actually written by Carl Walters and Howard Odum. Later in 1976, H.T. Howard Odum published a book, Systems Ecology, and the subject came to limelight. And that book is considered as the Bible of systems ecology. Okay, but the actual scientific applications were realized after Rio Summit in 1992. I'm just a brief history I'm telling about that. So this is the picture of his elder brother of HT. He is Eugene P. Odum. So he was in the Odum School of Ecology, but HT was in University of Florida. So Odum students, Mark Brown, University of Florida, David Tilley, Patrick Kangas, University of Maryland, Daniel Campbell, Environmental Protection Agency, Sergio Ulgietti from Italy, University of Siena, Robert Costanja. He was in US, but now he is in Australia. William Mish, he was in Ohio State University, now he is in the University of Florida. And some other renowned scientists, like last week you have heard the lecture, Barnard Patton, Sven-Erik Jorgensen, and Robert Ulanowicz, developed systems ecology in different branches. And Sven-Erik Jorgensen, he passed away during 2016. Sven-Erik Jorgensen, he is also my teacher. Sven-Erik Jorgensen also interlinked all the branches of systems ecology. So from systems perspective, several theories are developed, and these theories are called the integrated ecosystem theories. And these are mainly based on thermodynamics and information theories. Okay, now, next I'm coming to different branches of systems ecology. So first is ecological modeling. That is, I told you that without mathematics, different types of mathematics, you cannot penetrate the systems ecology. So the dynamic modeling. Dynamic modeling, there are two types of dynamic modeling, in temporal scale or spatial scale, or maybe spatio-temporal scale. So whatever the system is at this moment, what will be the system after 1 year or 1 month or 10 years, something like that. This is the dynamic modeling. And structural dynamic modeling, almost dynamic modeling, but here the more complicated one, structural modeling, it needs optimization. And the static modeling. Actually, static modeling is a tool which can deal the whole system, not the dynamic and structural modeling, structural dynamic modeling. So Sven-Erik Jorgensen is a pioneer in developing dynamic and structural dynamic modeling in systems ecology. So during 70s, he published a book that is Ecological Modeling, mainly based on the dynamic modeling. That is also considered as a Bible of the dynamic modeling. And Barnard C. Patton and Robert Ulanowicz are the pioneers who developed static modeling. I will discuss, whenever I will discuss static modeling, I will discuss about their contribution. Okay. Now, I'm coming to the dynamic model first. Okay. So dynamic model may be theoretical and database simulation model. So theoretical model actually does not require any data, only need the conception. From the conception, you can go through some mathematical thing, and obviously dynamic model, those who are the dynamic modeler, particularly theoretical modeler, they go for two types of solution, mainly the analytical solution, and it needs a lot of mathematical skill. So like us, we are actually from biological background, it is not possible for us for the analytical solution because it needs a lot of theories, lot of lemma, and etc etc. So no software is developed to go through the analytical solution. Only you can, with the help of software or help of program, you can go for the numerical solution. Numerical solution means you need some numerics, the real data. So sometimes you can use the numerical model with the help of the arbitrary numerics, but generally the theoretical modeler they do. I also did some work. I will show you that. So what are the major elements of the dynamic modeling? The state variable. State variable means which varies and which actually uh, the state of the ecosystem. Suppose I'm telling one model. Suppose phytoplankton, zooplankton, and fish. So there are three state variables. Here is the phytoplankton, zooplankton, and fish. Something like that. And forcing function are external variables. Sometimes some function, suppose sunlight. Sunlight actually forcing the behavior of the phytoplankton, or the temperature, nutrient. So these are the forcing function, how these after their forcing nature, how the state variables will behave, that will detected by the forcing function, and then ultimately the whole thing can be converted to mathematical equation, and whenever it is converted to mathematical equation, then it is called the mathematical model. And unless and until you convert the whole conceptual model into mathematical model, you cannot handle the model. And the parameters. Parameters means here we consider generally you, the biologist people, you use the parameters in a different context. We use the parameters in rate parameters. What is that? Suppose, what is the rate of photosynthesis? What is the rate of zooplankton grazing on phytoplankton? What is the rate of death of zooplankton or death of phytoplankton? What is the senescence rate of phytoplankton? What is the predation rate of fish? What is the migration rate of fish from here to other? What is the catch rate of fish? Something like that. All the rates are called the rate parameters. And universal constant. Generally, most of the dynamic model, in case of parameters, we consider parameters is of the constant value, but actually parameters is not the constant. Actually, in reality, is not constant. Suppose the photosynthetic rate is not constant, but for the simplicity of the model, we consider is a parameter is a constant. But in case of structural dynamic modeling, parameter is also considered as the variables. That's why it's very, very complicated. I will discuss later on. And then the universal constant. Universal constant, sometimes in the equation, you have to take some, the constant, gas constant, or the atomic weight, or something like that. So these are constant, wherever you do the modeling. Now, I'm coming to the theoretical model. Just I'm going, then I'll go to the case studies of theoretical modeling. So suppose this is a simple model, but is a system. And I'm not telling this a whole system, because other than phytoplankton, you can say like that bacteria is there, or fish can be small fish. There are different types of fish, planktonivorous, detritivores, or something, omnivorous fish, something like that. Suppose there is a system. This is a theoretical model. First is the phytoplankton, the first topic level, then the zooplankton, and then the fish. So phytoplankton is taking nutrients or energy from the sun, and then that energy is moving to zooplankton through grazing. Whenever they feed, zooplankton feed on phytoplankton, then actually energy is moving from phytoplankton to zooplankton, and from zooplankton, then the energy also moving to fish whenever the fish predate on jade. So this is the simple, very, very simple conceptual model. Now, if we consider this one into some mathematical one, So this is the ordinary differential equation. So this is a temporal scale model. Okay. Now little bit I'm telling about the, I'm not only the equation I will show here, not other cases I'll show an equation. Suppose phytoplankton, and this is dp dt, that is in terms of when time is changing, phytoplankton is changing. So this is, that part is a phytoplankton growth rate or the phytoplankton growth, and you see this is a plus, no sign means plus, and this is minus. Minus means, so next is the zooplankton. So zooplankton is eating, grazing on phytoplankton. So that part is getting deducted from the phytoplankton, phytoplankton component. So that part actually, this is a type 2 type of equation. So A1 is the, the grazing rate of zooplankton on phytoplankton. That's why Z predating or rather grazed on P at the rate of A1. And so these are type 2 equation. The most of the biologists, we call is the Michaelis-Menten equation. So we call is the Holling type 2 equation. Okay. And obviously the equation first actually proposed by Holling, but Michaelis-Menten actually used that equation in the enzyme kinetics. Anyhow, so now the question is, so that part is deducted. Suppose if that part is added, not exactly same amount is added, for the theoretical model, for the simplicity, if we think like that, that part is added to the zooplankton. So zooplankton energy is coming from that part, and ultimately that is minus. Minus means the fish also predating on zooplankton. So that part, so A2 is the predation rate of fish on Z, and this is also Michaelis-Menten or the type 2 equation, and this is the zooplankton mortality rate. And now that part is added to fish, and that energy from the zooplankton is coming to fish, and that is the dynamics. Due to that, the fish dynamics is depending, and now this is the mortality rate of the fish. Okay, so this is a very simple equation. Now you can ask me, what is theta? So why the theta came? Just I'm little bit discussing why the problem came to me, or came to my mind, and I actually formulated the model, and actually one of my students actually, he did his PhD particularly in this model. So generally the basic model is proposed by a very good mathematician, so Alan Hastings. Now he's in University of California, Santa Barbara, and we have modified that model. That is A1 minus theta. What is theta? Actually, we have seen that generally, whenever the system is normal, the system is the equilibrium state. That means equilibrium state means neither any of the component, either P, Z, or F, they are not oscillating. That means they are, they are in time bound. They are neither increasing nor decreasing. But generally, what happens? Suppose if Z zooplankton predation rate, grazing rate, A1 is increased. So if A1 is increased, then that part is actually more. So in that part is more means zooplankton will grow more, and if grow more, then that part will be more, and ultimately the phytoplankton will be decreased. Phytoplankton will be decreased. So whenever the phytoplankton will be decreased, the Z also will be decreased. So in that case, the oscillation starts. So whenever the oscillation starts, if still it is increased more, and ultimately the system will be in chaos. So whenever the system is in chaos, the system can be extinct anytime. And this is a Kaufman most important theory. The system is self-regulatory. The system is self-controlled system. So in that case, what happened? There are different types of phytoplankton in the system. Some of the phytoplankton, they produce toxins. So that type of phytoplankton, they are actually, their number in the system is very, very less. But whenever those planktons which are not producing toxins, if these planktons are eating more, that means they will get more space. That these toxin producing phytoplanktons, they will get more space and they will increase more. So in that case, so the, you know, so the toxin, if you consider the theta is the toxin, so if toxin is more, in that case, the zooplankton will feel some problem, and in that case, their A1, their grazing rate will be decreased. Okay, so if you increase, if theta is not there, if you increase, if A1 is increase and increase and increase, then ultimately the system is in chaos. But during chaotic system, the system is a self-organizing system. The toxin phytoplankton produce theta, that is toxins, and whenever the toxin is produced, then A1 is getting decreased. And in that case, again, the system is moving back from chaos to the normal one. And ultimately, the system will be again in the equilibrium state. So that's why we have proved from the theoretical model, the system actually is a equilibrium system. I will show you through graphs, then it will be clear to you now. Okay. Suppose, just little bit going to the results, I'm telling about something else. So there are, this is oscillations. Oscillation means, suppose if it is x is a component, biological component, suppose. So it is increase, then decrease, increase, decrease, increase, decrease. But in terms of some time, so what will happen if you increase the time? Then the uh, oscillation range will be gradually decreasing, and ultimately the oscillation stops. Where the straight line will come, the oscillation stops, means the x is neither increasing nor decreasing. So this type of oscillation is a convergent oscillation. And convergent oscillation is very good for the system, because the system was in turmoil. Initially is huge turmoil, and ultimately getting less and less, and ultimately the system is in order state. So reverse one, in case of divergent oscillation, the system is in the order state, but the oscillation starts. That, ultimately the more and more time will go, the system will be more oscillates, and ultimately what will happen, sometimes that downward trend, it will cuts to the 0 x-axis, and whenever it cuts to the x-axis, the x will be 0. So if any biological component is 0, then it will never come unless and until migration from the other system. So divergent oscillation is not at all good for the system. Okay, so now what is the equilibrium state? Suppose x, y, z. Suppose x is the phytoplankton. So in response of time, whatever the phytoplankton is time t0, and whatever the time tn, the phytoplankton is neither increasing nor decreasing. So according to bottom-up effect, zooplankton is depending on x. So therefore, their population is not increasing or decreasing. If zooplankton increased, then x also will be decreased. And if it is decreased, then x will be increased. So if one shows the equilibrium state, the higher trophic level also shows the equilibrium state. And even the Z also means here the fish. Suppose fish also will show the equilibrium state, but their equilibrium point is not at the same scale. So what happened in case of ecology books, we actually called is the equilibrium point. So if you put the three-dimensional graph, suppose phytoplankton, zooplankton, and fish, then it will be in a single point. That point indicates the amount of x, that point indicates the amount of y or amount of z. So this is the equilibrium point. So generally in a good system, the system is in the equilibrium state. Okay. Now, if system shows oscillation, if x shows oscillation, so then y also will show oscillation. So z, that means if phytoplankton shows oscillation due to bottom-up effect, the zooplankton also shows oscillation, and the ape also shows oscillation. In the other way, if fish only shows the oscillation, if fish shows the oscillation, this due to the top-down effect, the zooplankton also shows oscillation. If fish is more, then zooplankton will be less, something like that. So in that way, so if the oscillation is something like that, there is a upper asymptote level and the lower level also. It will never come to the that particular level, or never up to that particular level. So in that case, if you put the three-dimensional figure, then it will show a cycle, not circle, it's a cycle. So that particular thing is called the limit cycle, because the oscillation within the limit, the upper and lower limit. So this is the limit cycle. Okay. Now, next is something like that. One oscillation is high, then the smaller oscillation, then high, then smaller, then high. So in that case, so all will show the same type of oscillation, and ultimately in the three-dimensional plane, you will get so this type of figure. This is called the period doubling or phase doubling, something like that. If the bigger oscillation, just you follow my cursor. So this is the bigger oscillation, then it will come to the smaller oscillation, and then it will come to the bigger oscillation. So simultaneously smaller and bigger. So this is called the period doubling. But now, if suppose in case of our study, theoretical study, if the theta is in, if the the tox, if the grazing rate A1 is increased, so in that case the oscillation will be something like that. So there will be no pattern of the oscillation. So one pattern of one cycle never repeat the subsequent pattern of any future oscillation. So this type of thing is called the chaos. So one pattern never repeat the pattern of the future pattern, future oscillation. So in that case, so this is called chaos. And if you put the three-dimensional diagram, so then you will get this type of figure. But sometimes the chaotic pattern, chaos, so the patternless oscillation, but so far the whole chaos is concerned, the chaos having a pattern. So sometimes is the teacup chaos or the Lorentz attractor like that of English 8 or Bengali 4, something like that. Now I'm coming to my work. So now what I told you that, so whenever the zooplankton grazing rate is normal, so then we have found that whenever the system starts, the system showing the higher oscillation, then gradually in terms of time, the oscillation is getting down, and ultimately it will come to the equilibrium state. Okay. So this is fish, this is zooplankton, and this is phytoplankton, all in the equilibrium state. And from this three-dimensional figure, so what amount of phytoplankton is here from this scale, what is the amount of fish, what amount of Z, you can easily calculate all these things. Okay. Now, if A1, that is the zooplankton grazing rate, is increased, then the same system, whatever it showed the equilibrium state, now it is in the limit cycle. So now for this cycle, so the fish varies, P varies, Z varies. So if A1 is further increased, then it will come to the period doubling stage. And if it is more further increase, A1, then the system will go to the chaos. And this is a typical pattern of the teacup chaos. Just like a teacup, this is the handle of the teacup. And so this is the teacup chaos. So we have observed in many situations, there are 15 or 16 type of chaotic pattern. Actually, we have contacted a person who is working on chaos in Department of Mathematics, University of California, Berkeley. So actually, he also told like that he never seen that type of chaos also, but some of the chaos are identified, some of the chaos are not identified, but we haven't yet published this particular thing. Okay, now the question is, whenever the chaos will come, according to Kaufman theory, the system is self-regulatory. So during that time, the toxin phytoplankton are not grazed by the zooplankton, then the toxin phytoplankton getting more space, and they will increase more, and they will produce the toxins theta. So whenever the toxins is produced in the system, then zooplankton will feel some trouble. So there A1, that is grazing rate, will be decreased. So whenever the grazing rate will be decreased, then that chaos stage is moving back, transform into rather period doubling phase, or the so this is a period doubling, but not period doubling like that of the previous one. But now it again, if theta is more, then A1 will be further reduced, and ultimately it will come to the limit cycle. And again it is more theta, then the system will be again in the equilibrium state. But remember, in previous case, the equilibrium state was here, something like that, and now the equilibrium point is here. So it is a convergent oscillation, and ultimately the equilibrium state is here. So now the system is recovered from the chaos. Just like that, if you are in a disease, suppose it is in a chaos, but if you are whenever you are recovered from the disease, then you will be normal. But that normal you cannot compare with the normal before the disease. So after certain time, maybe the equilibrium point may be shifted from here to the original position or in the better position. We do not know. So this is the thing we have modeled in our theoretical particular part. This is one of the case studies. I'm not going to that in detail. Okay, so if you are interested in these two publications, so whatever I told about the case studies, that is the publication Order to Chaos and Vice Versa in an ecological system, and investigation of thermodynamic properties in an ecological model developing from order to chaotic state. And my student Sandeep Mondal, now he's in a good position in ICMR. He's most successful student of me. So he's from physics background. And now I'm coming to the another part. So just a minute. Okay. Now I'm coming to Now I'm coming to dynamic modeling, but here not the theoretical dynamic modeling. Here I'm coming to, so it is called the simulation dynamic modeling, or rather database dynamic modeling. So data means here not the arbitrary data. Here data means mainly the realistic data, the data which are collected from the field. Most of the cases, the modeler, they do not collect the data. The data is collected by one group and the model is doing by the other group. But in our case, most of the data, particularly for the simulation dynamic modeling, we collect our own. But obviously, all data are not possible to collect. And in that case, no modeler in the world, they collect all the data, because some of the data are missing. And without a single missing datum, you cannot run the model. So in that case, there are different procedure. So I'm telling one by one, not in going to the methodology part too much. I'm going to my case studies. So little bit I'm telling about the modeling part. First is the definition of problem or the definition of the system. So if you know what is your problem and what you have to do, then you can make a model. The conceptual diagram you have to make. Suppose conceptual diagram is very, very important. The people think like that. In my students, they feel like that conceptual diagram is very easy. You can make within 2 or 3 days. But generally, conceptual diagram can take 2 to 3 years to make the actual conceptual diagram. And actually, what things is important and what things is not important, you have to consider in the model. So what thing is not important, you will not consider that in the model, because the model will be very, very complicated. And model is the actually picture of the reality, but it cannot replace the reality. Okay, just I'm telling some example of Professor Sven-Erik Jorgensen. Suppose if a model of sheep, SHIP, sheep. So one person is making a model of sheep, how to make a body of the sheep which actually minimizes the what I can say, interaction with the water. Minimum friction will take place. In that case, the shape of the sheep is very important, weight of the sheep is very important, but not the color of the sheep is important, or the cabin layout is not important, but how much electric wire is required to actually to produce the electrical line inside the ship. In that case, the cabin layout is important. Maybe the shape is also important, but not the weight is important. So what type of model, what is your problem? According to, you have to make the conceptual diagram. And generally in the database simulation modeling, almost all possible important things are included in the model. That's why the equation, differential equation, or more order differential equation will be formed in the model. And as well, all the equation, every order is very, very complicated one, because you have to consider many, many things. So in that case, analytical solution is never possible in case of simulation dynamic modeling. Only the numerical solution is possible with the help of computer, because with the advent of computer, the development of the computer, actually the numerical or the simulation, dynamic simulation modeling is nowadays is a powerful tool in any type of biological science or in the physical science too. So these are the steps. Then the conceptual diagram, then that conceptual model is transformed into mathematical model, and side by side you have to collect the data, because it's a database model. So according to your data, sometimes you have to change the conceptual diagram, and if you change the conceptual diagram, then you have to change the mathematical formulation also, and ultimately the sensitivity analysis. So what will happen, I am telling you, mainly the rate parameters, so suppose the predation rate, grazing rate, mortality rate, these rate parameters data are not always available. Some are missing. So the missing data, without any missing data, you cannot run the model. In that case, what will happen? We collect the data from the other system, the almost same type of system. If you get it, so then some average value we can use in that one, and you can run the model. And during running the model, actually we will go through the sensitivity analysis, and sensitivity analysis, some of the rate parameters, 2 or 3 or maybe less, so they will be more sensitive. So we have to handle the sensitivity analysis. And then on the sensitive parameters, if these parameters are unknown, rather not collected, then we have to calibrate that particular thing. So this is the second process, the calibration. And if it is collected from the field, so it cannot be calibrated, it is a normal data. But if it is not collected from the field, then you have to calibrate it, which particular value is more suitable for your model. Then that particular process is the calibration. And then whenever the calibration value of the rate parameters or the rather unknown value or unknown data is available, so then calibrated data, then after that you have to run the model, and you have to validate the model with the observed data. So whenever it goes successfully through calibration and validation, so then we can say the model is very realistic, and the model can be used for any purpose. So I'm coming to some case studies. Okay, so one of my major field is actually Sundarban mangrove ecosystem. Actually, I'm doing, I have been doing for the long time. I have been doing for the long time in the Sundarban mangrove ecosystem. And in the Sundarban mangrove ecosystem, actually, I work mainly on the water, actually aquatic system. So my field is here in Sagar Island. Initially, I did my PhD from here also. And also, so this part also I have collected from that part data, but that part, the data is very difficult to collect, because you know that that part is a very, very dangerous part, because tiger infested area, and the core area, the government will not give you the permission. Generally, we collect the data from our own, also from the secondary data, also depending on. Now, little bit dynamic modeling, dynamic simulation modeling I'm telling. So one of my student, he was awarded PhD thesis with this particular dynamic simulation modeling on nitrogen cycle. What is nitrogen cycle? Here you see like that. So the diagram is very complicated. So this is the liter biomass, and ultimately in the liter biomass, so in the liter means from the liter from mangroves, and then the nitrogen fraction of the liter, and ultimately it will come to the system and in the soil. So there is a state variables. So these the 1 2 3 4 5 6 7 ordered equation and differential equation, and then soil total organic nitrogen, soil total nitrogen, and soil total nitrogen is again divided into two, that is soil organic nitrogen and soil inorganic nitrogen. Okay, suppose so there are many there are many forcing function here. It is forcing the conversion from soil total nitrogen to soil organic nitrogen, and again soil organic nitrogen, so some part of soil organic nitrogen is lost. So this is the loss syndrome, actually sign. And now the soil organic nitrogen is again transforming to water, not untransforming. That is, some part of soil organic nitrogen is actually mixed with the water, or rather drained to the water, and this is called the ultimately in the water is, we named this as a water total organic nitrogen. And then water total organic nitrogen, or rather soil organic nitrogen, is also some part of the soil organic nitrogen is transformed into PON, particulate organic nitrogen. We consider it as a detritus form. Okay, and now again water total nitrogen is also come to the particulate organic nitrogen, and now this particulate organic nitrogen again transform into dissolved inorganic nitrogen. Here also the water total nitrogen is transformed into dissolved organic nitrogen, and dissolved organic nitrogen into transformed into dissolved inorganic nitrogen. Here also soil organic nitrogen is transformed into soil inorganic nitrogen, and ultimately transformed into dissolved inorganic nitrogen. You know that these are all the bacterial process, mainly the bacterial process, or the fungus, bacteria, and some insects are also involved. And this is called the decomposition process. And what is decomposition process? Decomposition process is a process by which the complex organic matter is transformed into simple inorganic matter. So this is actually scientifically this is called decomposition. Decomposition means complex organic matter is transformed into simple inorganic matter with the help of some bacterial or fungal or some other degradation. So the things, the nature is something adapted in such a way, the planktons or the plant, whatever it may be, they can only take the inorganic form of any nutrient, not the organic form, unless and until this organic part is transformed into dissolved inorganic nitrogen and mixed in the soil or the water, ultimately in the water. The plant, means phytoplankton, minute plant or larger plant, they cannot take is as a diin. So our aim in that particular study was like that, how much liter biomass is transformed into DIN, and how, rather in other way, how DIN is depending on the liter biomass. We have done through this very, very complicated modeling, and one of my student, Dr. Sudipto Mondal, now he is in the environmental science, University of Bardwan. He actually he did that work in his PhD thesis. Okay, now, so in case of sensitivity analysis, so you see lot of sorry, you see lot of parameters we have taken, and which parameter is more sensitive. You see here the parameter is a sensitive one. Sensitive is the green color. You see here green color, and most sensitive parameter is here. That is loss rate of humic acid and fulvic acid from SON to the system. So if I go to the diagram, so that part. So that is more important part. So if huge amount of loss due to that, then the DIN will be very, very less. So DIN is depending mainly on the liter biomass, but so far the internal mechanism is concerned, that part is very, very important one. So that part. So in that case, from this particular modeling, we have calculated that particular part is very important, and whole dynamics is mainly, if that part is little bit affected, then whole system will be in jeopardized. Okay, so now whenever we have started the thing, you see, So there are two phases. So what are the phases? One is the calibration and is the validation. Calibration means the whatever the rate parameters or rates are not available, that rates we have calculated through some procedure, that is called the calibration. So whenever we calculate the calibration procedure, so the green color is the observed data. So this is the soil total nitrogen. We have collected monthly data, and the red colored is the collected data, and the same, so initial data, suppose the same data we have put in the model and we have run the model, and model will give the results for the same year. So whenever the model results is actually the blue one, and blue one whenever matched with the red one, that means our model actually predicting the data, whatever we have collected the data same year. So we have, whenever we have run the model of the same year, then the same type of results is found also in the observed data. So this is called, so this first, so 1 year data we use for calibration procedure. Then we have changed the data, but the rate is same, rate parameters, that value. And whenever change the data, then the second year data we call is the validation part. So whenever our model pass through the calibration and validation phase, so we have go through the goodness of fit test, how much these particular two are matching with each other. So then it can be a, the test for what type of model results are matching with the observed results. So this is one of the state variables. So we have 7 ordered differential equation. So next is the soil inaugurating nitrogen. Suppose here also you will see the first year data, there is the calibration procedure, and the second year data, something DON, and so the observed DON, dissolved organic nitrogen, dissolved inaugurating nitrogen. So every state variables we have calibrated and validated. So soil total nitrogen, soil organic nitrogen. So this is the validated data. Previous one is the calibrated one, and ultimately we are passing through the calibration and validation, and every point every for every state variables we have gone through the goodness of fit test. And whenever goodness of fit test is significant, then we can say like that our simulated results are matching with the observed results. Then we can say the model is perfect model or the realistic model. Now you can ask me, so if you have the observed data, then what is the utility to have the simulated data? Many people, many students, they ask me, sir, then what is the utility of the modeling? Now I am coming to the you know coming to the conceptual diagram. Then I am telling what is the utility of the model. Suppose whenever the model is perfect one, the model is very realistic after calibration and validation. Now if you say like that, say suppose if liter biomass is decreased 20%, so just you make the liter biomass here, make the 20% decrease here, and just you run the model. So 20% if you decrease the liter biomass, then it will reflect the DIN. So what will be the DIN loss? So DIN will be 20% reduced, or DIN will be 80% reduced, or DIN will be not very much affected. So all these things, or something like that, if you suppose if that loss is 5% is less, then the liter biomass is same, that loss is 5% less, then what will be the effect in the DIN, or what will be the effect of the other? That can be easily within a minute you can calculate with the help of this model. That is the utility of the model. Unless and until you make the model realistic, then you cannot handle the model. Whenever the model is proved it is realistic model, after passing through successful calibration and validation, then you can say the model is very realistic, and the model can be utilized for any type of prediction or any type of utilization. Okay. Now, the summary of the nitrogen cycle, whatever my student did. So, contribution of DIN to the Hooghly estuary is dependent on high liter production. So, if mangrove is lost, in that case, the productivity of the estuary will be very less. So, whatever the good fishes, we are the Bengali people, we like fishes. So, lot of the brackish water fishes are coming to Calcutta, suppose Parshe, Bhangor, Topse, etc., etc. So all the fishes are coming from the Hooghly estuary. That that fish will not be available in the market if little bit litter biomass is less. Then soil pH and soil salinity are considered to be the key factor for the conversion of STN to SON, whereas redox potential plays an important role in the conversion of STN to SIN. So redox potential, I am not going to the chemistry, just I am going to the observation. I am predicting all these things through simulation modeling. So redox potential and conditions of soils are important. Mineralization of SO2, SIN is governed by microbial activity, etc. Dione degradation is governed by hydrolysis. Dione dynamics of estuary depends on mineralization. And loss rate of humic acid and fulvic acid from SON is most system sensitive parameters. So this is the most regulating parameter of the nitrogen cycle in the mangrove and estuarine system. Leaching rate, etc. I'm not going to the detail of that. Now, so these are the publications. So I'm not going to the detail of that. So this is the publication of nitrogen cycle. My another student, Joita Mukherjee, she did her PhD on carbon cycle on the same system. So you can go through these two papers and you can get the good picture of the nitrogen and carbon cycle of the Hooghly estuary. Okay, next I'm coming to the structural dynamic modeling. So structural dynamic modeling here, actually I did that work long back. So that is the data, whatever I used here, that is the used in the Central Europe. During that time I was in the Czech Academy of Science, and during that time I did that work. So structural dynamic modeling is very, very complicated one, because what I told, suppose I told you that the phytoplankton photosynthetic rate. Phytoplankton photosynthetic rate is actually considered the average rate in case of dynamic modeling. But structural dynamic modeling, so the rate also varies. There is a minimum and there is a higher limit. Okay, so in that particular range, any value can be added. Now, every rate parameters, these are the variables. If you consider these are small variables, so then just imagine all the parameters are interacting with each other. And in that case, so any value can be combined with any combination of values. So thousand combination will be there. And you have to run the model thousand times. So it is not possible. So in that case, so if you have the goal, if you have a problem and if you have the goal, suppose if your goal is in particular, so this is a model, just I'm telling first the model, the nutrient, phytoplankton, zooplankton, small fish, large fish, and their dead body constitute the detritus compartment. So this is the simple model. Now the question is, so in which type of the best interaction will be found where the F2 large fish production will be more? You do not know. But with the help of the structural dynamic modeling, if you put the ranges of the rates, in that case, particularly if you put the what is the largest value of F2 will be obtained, you can optimize the thing. So in that case, it needs the optimization technique and lots of, even in this particular model, sometimes in case of eutrophication condition, 4 days, 5 days you have to run the computer, then you will get the results. It is so complicated in a modern computer also. Just I'm telling about. So these are typical. I'm not going to the detail of the mathematics. Is a very complicated one. So suppose the phytoplankton, this is the scale of phytoplankton, this is the scale of zooplankton. And phytoplankton, so we have taken the phytoplankton body volume. So body volume in log scale, so it is 10 mu m cube, that is log 1, and 10 to the power 7 mu m cube, several meter long algal filament. It may happen like that. So their body volume varies something like that. So zooplankton body volume, so this is also 10 mu m cube, very small, cannot be visible on a naked eye, and 10 to the power 4, very large zooplankton. So what happened? Actually, I have gone through a book. Actually, that idea came from Professor Milan Straskova, Professor late Milan Straskova. He told me that, So you go through that book. So Implication of Body Size in Ecological Science, that is written by Peter, Cambridge University Press book. In that book, it was written like that. According to body volume, the rate parameter varies. Suppose I'm telling you the, the respiratory rate of, or rather the heartbeat is more in case of child rather than old person. So something like that. Even the heart rate of mice is much more higher than the heart rate of elephant. But some of the rate parameters are actually inversely proportional to the body volume. Some of the rate parameters are directly proportional to the body volume. Suppose the half saturation constant is directly proportional with the body volume. So in that case, if you know the body volume, which type of body volume, minimum or maximum body volume of phytoplankton is present in the system, then you can easily, with the help of regression analysis, you can easily calculate according to body volume how the rate parameter actually depending the regression equation. So in that case, if you put the differential equation, you put the regression equation in the differential equation. So if you change the body volume, then according to body volume, all the rate parameters of phytoplankton will be changed. According to in the zooplankton equation, if you put the regression equation of body volume, then according to regression equation of the body volume, the rate parameters also will be changed. So in that case, so thousand and thousand combination of zooplankton and phytoplankton will be found. So suppose if you increase 10 to the power mu m cube, 10 to the power 1 0.1 mu cube, 0.2 mu m cube, something like that. So in that case, lot of variation will be found with all the parameters. So if you optimize the thing, here we optimize the ascendancy. I will discuss ascendancy later on. So in that case, so this is the oligotropic, less nutrient condition. So in that case, where the ascendancy value will be more. So here, so that means in which combination of phytoplankton and which combination of zooplankton. So then if you know that combination, so that particular body volume, what are the rate parameter values through regression equation, and if it is here, so what are the through regression equation, what are the rate parameters of zooplankton, then these particular rate parameters combination in the oligotropic condition, maximum ascendancy or maximum fish production suppose will be obtained here. So in that case, structural dynamic modeling it can be done. Suppose if you go to the mesotropic condition, you see here it was here, but here now shifted from here to here. That means the zooplankton size is changed as well as the phytoplankton size is also changed. So in whenever the little bit condition of the nutrition is better, nutrient is better in the system, then the ascendancy value will be in the maximum ascendancy value in the different combination. So in that case, different combination means in different body volume, and different body volume mean in different rate parameters. So in which rate parameters maximum ascendancy or suppose maximum fish value is fish biomass is present, that can be easily calculated from the structural dynamic modeling. Suppose in the eutropic condition, you see the graph is shifted here. That means maximum ascendancy value is here. That means here the phytoplankton is slightly changed. The zooplankton body volume is actually selected here more. That means rate parameter is more changed in zooplankton rather than in phytoplankton. And ultimately, so different condition, if you optimize the thing, then you can get the actual value according to your, if you want the particular goal, and which value the goal can be obtained with the help of the structural dynamic modeling, it can be easily calculated. Okay, so this is very complicated one, and very few structural dynamic modeling has been done. Actually, this is the second structural dynamic modeling. I first used the structural dynamic modeling, that concept I did that one with the help of Professor Sven-Erik Jorgensen. So whenever I was in his lab, he told me about the concept of structural dynamic modeling, and then I first produced the structural dynamic modeling with him and Professor Milan Straskova. And ultimately, very few structural modeling till now have been done by the people. So people are not much more interested in the structural dynamic modeling because it very difficult to get the data, very difficult to execute the structural dynamic modeling. Okay. So if you are interested on structural dynamic modeling, so this is my two papers. So one is in the Journal of Biological System, World Scientific Publishing Company. Another is on ecological modeling. Okay, now I'm coming to the static modeling. So static modeling, actually, if you study the whole system, so whatever I told about the dynamic modeling, theoretical one, simulation dynamic modeling, or the structural dynamic modeling, these are not the whole system. These are the part of the system. But static modeling is more or less is the whole system. But still, it is not whole system, but it's a whole system. So there is a little bit history of static modeling. And static modeling, the main theme came from the um, a famous economist, that is Leontief. Leontief, maybe 1976, he got the Nobel Prize in economics. And during 1955, he produced an analysis, that is input-output analysis. What is input-output analysis? How much raw materials is required to produce the consumable goods according to the demand of the consumers. So for this, he produced the input-output analysis. Rather, first Professor Hannon produced the rather implement that input output analysis in the ecological science. So how much solar energy or raw material is required to make the biological components to construct the ecosystem, something like that, if you think in other way. And there are two major school in static modeling. So one school is actually uh, rather formed by famous modeler Professor Bernard Patton. Last week he delivered the lecture. His lab in the, or rather his school call is a network environment analysis. Actually, you have heard that I'm NEA last lecture. And another one that is Professor Robert Ulanowicz from University of Maryland. So he called is the ENA, Ecological Network Analysis. And I know Professor Patton and Professor Ulanowicz, they are very, very good friends. And naturally, they are giant in the static modeling. So most of the students now, whatever they are throughout the world, whatever they are doing static modeling, either they are student of Barney Patton or the student of Bob Ulanowicz, or maybe both, student of both. Okay. So ecological network analysis. Just I'm actually, although I was in the Bernie's lab, but I actually preferred the ecological network analysis, ENA. And what is ENA? So reasons to consider ecological network analysis. Dynamic simulation models, through useful, often have limitations because it is a 7-order equation. If it is suppose if there are 80 components, 80-order equation you cannot handle. So therefore, simulation modeling, dynamic simulation model having some limitation, not predictive of qualitative changes of ecosystem status, and some qualitative changes cannot be predicted to model using this approach. So these are the cons of dynamic model, simulation model. And need for understanding whole system properties, measure the flow of materials or energy. So in case of static modeling, handling a larger number of components, can work with comparatively limited amount of data, and evaluating the importance of each component. So ENA is based on a combination of 5 different related aspects. What are the aspects? One is the thermodynamics. I told about what is the thermodynamics. So everything is obeyed the law of thermodynamics, all the biological aspects. So statistical mechanics is a very important tool which is used in the ecological network analysis or the static modeling. So input-output analysis, I told about you. That is a Leontief original input-output theory and information theory. So a lot of information theories applications in the static modeling, and R taxonomic aggregation. I'm telling what is R taxonomic aggregation. Still, I'm telling you, even the static modeling have some limitation. That's why we have to reduce the compartment or the components. So sometimes we have to merge different components into a single components or the single compartment. So this is called R-taxonomic aggregation. So why R-taxonomic? Suppose, so one compartment is a name is a detritus. So detritus, or rather detritivore, suppose the component which is actually compartment which is actually eating on detritus, that compartment is the detritivore compartment. So detritivore, maybe the fishes are detritivore. Some fishes, suppose parse, mugil, parse, bhangor, these are the detritivore. Even some bacteria is also detritivore. Some fungus is also detritivore. So far the ecological function is concerned, they are put together in a single compartment. They are called the detritivore. So taxonomically, bacteria is far different from fungus. They are far different from the fish. So if you club together different components in a single compartment, although they are taxonomically are different from each other, so this is called the R-taxonomic aggregation to simplify the model, or rather to make the to avoid the complexity of the model. Okay, now just you think about the complexity of the model. So these are the, these are the compartments. Okay, so maybe the biological, maybe abiological also. Suppose the detritus is the abiological components. The question is, so but the network analysis is concerned, that particular compartments or components we call is the node. Okay, and one node is connected with the other node either directly or indirectly. So if node is connected, that connection is called the arc, arc. And arc is directed, that means arrow is there. So from where to where the energy or material is moving from which component or which node to which other node. So so these are directed arc. So now the question is, so there are, I'm not going to that later on, but so far the mathematics is concerned, particularly in case of static modeling. So this is the mainly the backbone of the static modeling is the matrix. So without matrix, you cannot proceed to the ecological network analysis or the static modeling. So to handle the matrix, those who are actually a little bit know about mathematics, if the matrix is 3 by 3, then you cannot solve in the calculator also, in the good calculator. So in case of ecological network analysis, there may be 30 by 30 matrix or something like that, or maybe 15 by 50 or 80 by 80 matrix. So there are a lot of softwares which are all freely available. So these are the software. If you are interested, then these softwares are available. And you can go through the particularly the static modeling. Now I'm going to the some little bit methodological part. Suppose there is a system, phytoplankton, bacteria, these are carnivorous fish, detrivorous fish, and these are detritus. Suppose these are particular simple model, and you see there are different types of arc. Whatever I told, this is the node, the 1, 2, 3, 4, 5 nodes. So this is one node, this is all the nodes, and all the nodes are connected by arc, either directly or indirectly. So this node is indirectly connected with this, because why? Because this node is connected to the detritus, and detritus is connected to the bacteria. So in that case, the bacteria is indirectly connected to the phytoplankton. And you see here the interesting thing, there are 4 types of arcs or 4 types of arrows. So one arrow is node to node, that is flow from one to another compartment. So this is node, this is another node, this is node. These are all in between the nodes. And another node is, so it is from outside to the node. So something energy or material are coming from outside to the node, and this is from node to the outside. So some energy or material are going from the node to the outside or the other system, and these are the nodes. So these nodes are actually the dissipation node or the entropy, particularly some part of the energy are dissipated. That means dissipated energy means energy is neither lost nor created. So that means that is a not lost energy. That energy is not give any further work. That's why it's the dissipated energy. So every time all the components are needing some energy, and during energy they are doing their work, and the work is done, and during work some amount of energy is dissipated. So that's why all time the energy is actually lost from the system, but not lost from there is a not workable energy. Okay, but that energy, whenever going outside, that will give the energy to the other system. So there are four types of flow: flow between the nodes, flow from outside to the node, flow from node to the outside, and flow from node to the dissipation. So the flow from node to node, so that is we call is a flow matrix. We create the flow matrix. I'll show you. And there are three vectors. Vectors is the z vector, means the input vector, that is from outside to the node, and export vector from node to the outside, and the respiratory or the dissipation energy, that is the respiratory vector. So now two school, I told you that the Patton school and Ulanui school. Patton school, they form the flow matrix like FGAI, that is flow from column to row. Okay, and in case of Ulanui school, they form the flow matrix, that is the inverse matrix. Actually, they form the flow from I to row to column. So I use the Ulanui school FIJ. Suppose here. So this is the first compartment, first node, suppose, and detritus is the fifth node. Okay, so if you number it, then so how many nodes are there? 5 nodes. So the matrix will be 5 by 5 flow matrix. Suppose the first node, phytoplankton, the energy is moving from the fifth node, detritus. So this is the fifth node. So detritus. So here flow from row to column. So row is 1. Okay. And what is the fifth node? Detritus. Detritus is the column, fifth column. So first row, fifth column. So you put the 2 here. Okay. Clear? Now the question is, suppose here, so detritus to bacteria. Maybe the bacteria is the fourth one. No, sorry, second compartment. We didn't put the name also. Second one, this is the fifth one. So now the no, sorry. Yes, no, sorry. This is the fourth row. So in that case, the fourth row and the fifth column. So in that way, so you put the 4, 9, etc., etc. In that way, you can put the thing, particularly. So no, so this is the second one. This is the fifth one. So the second row and the fifth column. So the 9. They put the 9, something like that. So in that way, or rather, from the fifth column. So fifth row, and this is a fifth and the second, and it moving from here to here, there is a 4. That is, this is the fifth row and this is the second column. So the 4 will be put here, and the wherever the data is not available, so we put the 0. So in that way the flow matrix will be formed, and Z matrix, that is the input matrix, suppose 1 to the 1 first one, and though no other input is there, only the fifth one is the input, so other is 0 and this is the 3. So in that way the same thing is there. Export one, the first one from 11, second one, so this is the 13, so fifth one is 12, something like that. So and respiration, all the thing 15, 16, something like that. So if you get the matrix, so then from the conceptual model you will get the matrix one. Now, so what things we do? So total system throughput, that is how much energy or material is coming and how these are utilized and coming out from the system, the whole thing can be calculated. That is called the total system throughput. And then the ascendance and development capacity, little bit I'm telling. So according to the TST, total system throughput, how much a system maximum developed, how much a system can be, that can be calculated with the help of development capacity. So C. So, but generally in reality, the system realistic performance of the system is actually ascendancy. So realistic development is always less than that of the theoretical development. So generally ascendancy A is less than that of the C. But theoretically, or rather if you argue it, then you can say like that A can be equal to C in some situation, but never happen, but can be mathematically can be equal to C. But generally in reality, A is always less than C. C is the maximum development theoretically, and A is the realistic development. Okay. And then the Finn cycling index. What is Finn cycling index? Finn, I told about the Thomas Finn. Thomas Finn is the most, according to Barney Patton, he told me Thomas Finn was his best student, best intelligence student. He is a chair professor of MIT now. Finn cycling index, that Thomas Finn. So what is Finn cycling index? Actually, whole food wave is a cycle, and inside the food wave there are many subcycles. So retention time of the energy or material is maximum in the whole system if many subcycles are present. That means Finn cycling index is very high or more than the other system. That means the energy will be utilized rather more proficiently, particularly in the system. So these are the things we calculate. And now I'm not going to very detail of that. So then up to 2009, most of the people, they thought like that if any system shows maximum ascendancy, then the organization and performance of the system is very, very good. But later, actually, most in our country, most of the things we found like that, the empirical researcher and the modeler, theoretical researcher, has some confliction. Empirical researchers, they think like that the theoretical, they do not collect the data, what they are doing, particularly the biologists, what they are doing nothing. And theoretical biologists, they think like that we are doing many complicated thing, you are just doing the normal work, normal routine work. But this is not the confliction. Actually, theoretical science is totally depending on the empirical science, and empirical science can also be developed with the help of the theoretical science. So whenever we believe like that, whenever the ascendancy is high, the performance and the organization is good, means the performance also good. So in that case, the system is very good system is healthy system. But later, from the empirical research, it was found that so whenever the system shows maximum performance, that system cannot withstand stress. That means the system is not resilient. Means the system cannot withstand stress. If any stress comes, the system may be degraded, maybe decayed, maybe lost, maybe extinct. So in that case, so that cannot be a good system. So average mutual information, I'm not going to that. Flow diversity, that means how much arcs are there, that is the richness of the arcs, and how much evenness of the flow in between the arcs, that two things are calculated with the whole help of flow diversity. Okay. Now, little bit I'm coming here. Coming here, the health of the ecosystem. Nowadays, health of the ecosystem is very important. It is very difficult to ascertain the health of a particular individual, human being. But how can you assess the health of whole system, several 100,000 individuals? So, but still, it is possible with the help of the systems ecology. So, healthy ecosystem is one that is sustainable. Am I right? It has the ability, these are Kostanjan, Maggie. So, it has the ability to maintain its structure, organization, and function vigor over time in the face of external stress, resilience. Kostanjan, Maggie, 1999. So, robustness, that particular concept came in 2009. Robustness, And robustness is depending on vigor, organization, and resilience. The organization, if the organization is very good, then the vigor also will be very good. That means if the organization is good, the system function or performance is also very good. But resilience, in that case, most of the cases, if organization is very high, vigor is very high, in that case, the resilience will be very less. So in that particular system, if any stress comes, the system can be jeopardized. So in that case, you have to consider the resilience. So whenever three things are very, rather adjusted in a proper way, then we can call the system is healthy. Next, I'm coming to the robustness of the system. Okay. So, okay, now, what is robustness? Suppose I'm telling about in this one. So I told you about the ascendancy. Ascendancy is the realistic performance of the system, and C is the theoretical performance of the system. I told you about that. A is always less than C. So if I put A by C, C is greater, uh, denominator is greater than A. So in that case, it will be some point value. So in that case, this is, we denote it as small a equal to A by C. And now in this x-axis, if we put A by C, what I told you that A can be equal to C theoretically. So in that case, if A equal to C, then small a value, that is A by C value, will be 1. So the maximum value is 1, and minimum value, if A is 0, then it will be 0, then the value is 0. So it is 0 to 1 scale. That is the degree of order. Okay. And now robustness is minus a log a. A means what is A by C? That means minus capital A by C log A by C. That means minus capital A by C log A capital A minus log C. So log capital A minus log capital C. So capital C is the bigger value. So in that case, that particular part will be minus. And A by C, if you divide and minus, it will be plus. So robustness will come. So you see here in the y-axis, you put the robustness value. So you will get, if you make the graph with different A by C combination, then you will get a inverted U-shaped curve. Okay. So that means whenever theoretically it is very high, equivalent to C, that means the organization, that is vigor, and performance, that is vigor, and organization also very high. In that case, suppose if A is equal to C, then the robustness will be 0. That means whenever it is 1, if A is equal to C, it will be 0. That means here the resiliency is 0. That means the robustness is also 0. That what will happen? If the system utilize maximum energy or all the energy for their organization and performance, then no energy is left to combat the stress. So in that case, the health is 0. That means the system is very, very bad. But at the same time, if A is 0, that means total energy is utilized to combat the stress. No energy is left for the organization and performance. In that case, it will also 0. So, but that is the middle combination. If A is a little bit half of the value of C almost, so then you see the curve is something like that. So that particular portion, this is called the windows of vitality. So if the robustness value, that means robustness value is very high here. So in that case, so the A by C combination value will be like this here. So in that case, all the three, what I told you, that the vigor, resilience, and other, that particular things will be maximum here. That is the windows of vitality. That means here you can get the more or less good performance of the system. That means good organization is the organized system. That's why it's getting the more or less good performance. As well, whenever the stress comes, the system can withstand the stress from outside, and ultimately the system can be very, very good system, healthy system. And this is my own example, just Just I always in my to my students also, I also give the same example. Suppose you have 3 cows. One cow gives 1 liter milk, another cow gives 6 liter milk, another cow gives 10 liter milk. Okay, and all the cows having the same amount of food, getting same amount of food. But now, which cow you will prefer? So before 2009, the concept in the ecosystem, actually we preferred the cow gives 10 liter milk and per day, because we during that time we always emphasize on the performance and organization. But if any disease comes, suppose stress, so the 1 liter milk giving cow, that cow is she is already in stress, so if any new disease comes, that cow will not be affected. So because that cow is habituated to combat the stress. In that case, that 1 liter milk will not be further decreased. So 6 liter milk giving cow, if any stress comes, there is some amount, that cow is more or less little bit habituated to combat the stress. So some part of energy is left. So for this energy, that cow can combat the stress. The 6 liter milk, that means the energy which is producing the milk, that energy will be allotted to combat the stress. So in that case, the 6 liter milk will be converted to 4 liter milk per day. But 10 liter milk giving cow, in that case, total energy is for the organization and performance. So no energy is left to combat the stress. The cow will die. So now, which cow is the most preferable cow? That is the 6 liter milk giving cow. So the system is something like that. If the system is highly performing system, highly organized system, highly performing, in that case, there is a chance the system can be degraded anytime due to stress. And stress is obvious. Any type of stress is obvious in the system, either man-made stress or the natural stress. So this is the actually concept of the ecosystem health. That is a finer balance between all these things. Okay, so this is the robustness. Now I'm the almost in the last end of my lecture. So this is the vigor. These are the things. NPP, net primary production, P is to R ratio, total system throughput, ecotropic efficiency. These are the indicators. Organization, the indicators are average mutual information, Shannon diversity index, development capacity, ascendancy, fin cycling index, A by C, etc., and resilience, redundancy, R by C, etc. These are the actually, I'm not going to the detail of that. So my case studies, I'm coming to that. So whatever the ecological network analysis we applied here in 2 systems, one is the mangrove system, another is the reservoir system. The mangrove system, I told about the map. I have shown already the map. And the reservoir system, my 2 students have already completed in the reservoir system PhD in the static modeling, and most of the part is done by Arnav Banerjee and his PhD thesis. And so far the Sundarban system is there. Nobbendu Rokhi is also have done, and myself during my student life, or even up to 2008, I did my own work in my own hand. So what I did here, so I took one food web, that is a benthic food web of the reclaimed island, particularly in the Sagar Island. There is 14 compartments here, and I published a paper with Bob Ulanowicz. So during 2001, in 2000, and ultimately we have shown that due to man-made degradation of the system, how much the system is affected in particularly the reclaimed island of Sundarban, particularly in Sagar Island. Initially it was a forest, but now the remnant of forest is there in the southern part of the forest, southern part of the island. That is the phoenix hetal in Bengali name. That particular tree is there, still little bit there. And later, in the mainly in the Lothian and Petis Island, I have collected some data, and I compared the same compartments I have chosen in the Virgin Island, and I compared the reclaimed with the Virgin Island, and I published one in ecological modeling that was published in Journal of Biological System. Okay, and now This is the part of the Bokresa Reservoir, whole reservoir, and these are the publication. A lot of publications came from that particular part in ecological informatics, ecological modeling. Even I did some work on South African estuary because my two students actually they visited South Africa for their study. Joyita Mukherjee and Arnab Banerjee. Although Joyita did work on the dynamic modeling, but after she was also postdoc in my lab during that time, she did some work on the static modeling, and both of the students, Arnab and Joyita, they did some work on the static modeling on um, Arnab did in Bakrasa Reservoir as well as on South African estuary. Joyita also did some on South African estuary too. Okay. Now you see that just I'm telling about. So this is the robustness of the pristine island that is in the Virgin Island in my study, and this is the robustness of the in the reclaimed island. So this is in the stress side. Reclaimed island is huge in stress, and even the Virgin Island that is on the almost on the edge of the windows of vitalities. But whatever the data we have gathered, the data is not exactly from the core areas of the mangrove forest. That is also in the stress condition, but less stress condition. So my idea is like that, or my assumption is like that. If we get some real core area data, so I think our the robustness index will be, or rather robustness point will be here, something here. So in that case, it will be in the Windows topmost position of the. This is the robust, more or less robust condition of the ecosystem will be found. So in case of uh, in case of the other system, particularly Joyita also did some work on that estuary, South African estuary, and also the Bokreshwar Reservoir. So we have calculated the robustness, and we actually perturb the system and how the robustness value is changing due to perturbation, that have also shown in that particular in these papers. If you are interested, you can get all these papers from my website also. Okay. Now comparative study of variation in ecosystem properties. One of my students recently awarded, Nobendu. Nobendu did a fantastic job, and he took two period of Sundarban. So one is from 1985 to 1990, another is 1980 to 2003, and he compared the two period of the Sundarban, same system, and how the system in time period, in time scale, how the system is affected. So just you think about that, sum up all consumption or something like that here. So it is less in the phase 2, but whatever our study is concerned, so not much changes we did actually observe in that particular system. But whatever the things we observe, the most important thing, most valuable thing, that is the fish, fish catch. Particularly in the fish catch, in the latter period, the fish catch is excessively increased, and if increase in that rate, then the system will be in real problem. So government should take some necessary action. But in our country, there is the most problem is the researcher is not actually connected with the government policy. But in abroad, in a good countries, developed countries, um, the researchers are very much connected with the government policy. So I think so the government policy should be like connected with the research work. Otherwise, what type of activities is necessary to conserve the system, to restore the system, um, it is not known by the government. And naturally, whatever the money is spending for restoring the system, that money will not be properly utilized. Okay. Now the things, the in the network analysis, we also found the keystone species, but this is actually a lot of problem in the keystone species. And keystone species is very important because their biomass is very less. But if you remove that particular species, then the whole system will be in jeopardized. So keystone species, there is a bug, or rather cons, in the ecological network analysis, because there are 3 keystone species, and 3 keystone species gives 3 different results most of the cases. But I think so none of the index is very fruitful for ecological network analysis. Okay. uh, I'm not going to that, because this is the Bakrasa River, is a majority estuary in the South African estuary. And if you are interested, then you can go through the temporal variation of keystone species. Regarding keystone species, all this paper is available in my website. And I'm not going to the, you know, rather extend my lecture more. Okay. Now, little bit I'm telling about some disadvantages. Whatever the advantage I told you about the ecological network, there are some disadvantages. ENA often falls short due to several limitations like non-inclusion of time factor. So, this is a static modeling. That's why it is not a dynamic factor. But nowadays, it is rather upgraded from static to dynamic model. So the software which is very useful, or rather utilized most in case of static modeling, that is EcoPath. Now from EcoPath, if you transform into EcoSim, then the static model can be converted to dynamic model, but it needs lot of data. And theoretically, I'm telling that this type of data is not possible. Even today, it's not possible to convert the static to dynamic modeling. And limited capability of result extrapolation. Networks are specific to ecosystem. Some facets are discussed here, particularly the path versus cycling. There is some problem. Critical critical arc versus weighted arc. I'm telling, sometimes, most of the cases we feel like that the arc having the value means some energy or materials are moving from one to another compartments. But generally we feel still now, the arc which containing more value, more flow is there, that card maximum flow is there, that arc is critical. But it's not like that. Sometimes the arc having, if you perturb that arc, that means that value can be flowed from through other arc to the other compartment. So the system will not much be affected. Many cases, but many cases it can be affected. Sometimes critical arc is something like that. If that arc is perturbed, that value cannot be flowed from that compartment to any other compartment. So, and if that arc is perturbed, the system will be maximally jeopardized. So in that case, we call it I call it the critical arc. And to identify the critical arc, that tool is not already developed, but nowadays a matrix, particularly matrix mathematics, a tool is developed very recently, that is in between centrality. And last year Nobel laureate Abhijit Binayak Banerjee, actually he did some in between centrality analysis. He is very good in mathematics, and particularly in the economics. In between centrality analysis, you can go through and you can easily calculate the critical arc. And I told about the keystone species. There are 3 particularly mathematical formulation by which the critical keystone species can be calculated, but 3 are giving different results. But system having only 1 keystone species. So I think none of the but which one is better, I don't know. So that can be corrected. And sometimes competing sub-food waves, ecotropic efficiency, these are the things. But actually, in one my lecture in maybe I cannot remember, maybe in France, in University of Toulouse. So during that times, I told about that, and Stuart Boret, my friend, actually he objected, no, you did something wrong, that's why you are telling these are the cons of the ecological network analysis. Later, whenever he did his work in his own field or own areas, he also realized that there is certain actually cons of the ecological network analysis. So during that time, so we developed some uncertainty analysis with the help of Stuart Boret. So in ecological network, uncertainty analysis shows how variability inherent in model parameters affecting model outcomes. and it uses linear inverse modeling, particular technique, statistical technique, to enable stronger support towards statistical data that are otherwise unexplainable through ENA, ecological network analysis. And that's why we actually named that paper Uncertainty Analysis for Ecological Network Analysis, enable stronger inferences. And that was published in a very, very good journal. It's a methodology journal, Environmental Modeling and Software. And so myself, Stuart, and Stuart PhD student David Hines, actually we produced that one, and we developed certain tool also. Actually, Stuart and his group developed, the R platform. So in R platform, you can do the ecological network analysis, and we have developed certain tool for this particularly inverse modeling method to answer for to identify the uncertainty so of this model. So you can go through the ENAR through the if you go through this paper, that particular software is available, freely available. Okay, now the acknowledgement. So my mentors, my collaborators, actually my mentors, one of the mentors, very, very good mentor, I can say, my philosopher guide, Professor Bernard Patton, and he's a doyen in static modeling. Professor Robert Ulanowicz, I have some publication with Robert Ulanowicz, but I only met him during maybe during 2000, during 2000 in University of Copenhagen. And in that particular conference also, Professor Patton came, and I met Patton first time also. So Professor Brian Fath, my friend, he's a very good student of Professor Patton, and now he's in the University of Towson, Towson University, and not University of Towson, Towson University, USA, and he is now editor-in-chief of Ecological Modeling, it's the best journal so far the process-based modeling is concerned. Stuart Boret, now he's a Professor Stuart Boret. Professor Stuart Boret, actually he's in the University of North Carolina, USA. He's also a student of Professor Patton. And David Hines, now Environmental Protection Agency, student of Stuart Boret. And Ursula Scarler, she was trained in the lab of Professor Robert Ulanowicz in University of Maryland. She is Austrian, but so far her job is concerned, she is in University of KwaZulu-Natal, South Africa. My student Joyita Mukherjee and Arnav actually visited her lab, and my students, they helped me a lot for the study. Joyita Mukherjee, Arnav Banerjee, Nabindu Rokhit, and another student is working with me for PhD, not Dr. Nabindu Rokhit now, and Sagota Sinha. So recently, she did publication on ecological network analysis, particularly static modeling, on the coupling phenomenon of pelagic and benthic food web in a shallow zone of Kakinada Bay. Okay, already it has been published in Ecological Informatics. Okay, thank you very much. Good night, everybody. Any questions? I will be ready to answer. It's my pleasure to answer your question.
Thank you, sir, for that insightful, wonderful talk. And we have a few questions. Should we go ahead with the questions, sir?
Sure.
So Shanchari Gorai is asking the question, in case of phytoplanktons, why the mortality rate is not deducted?
That is a good question. So actually, in the first model, you were telling about the theoretical model. Actually, I told you that this is a theoretical model. So everything is not possible, because if you consider everything, then it will be very difficult to handle analytically. This is an analytical model. And that's why the mortality rate, if you put the mortality rate, the model cannot be hampered, or rather model cannot be affected much. That's why we deducted that particular portion, the mortality rate. It's not so important, particularly in that model. That's why we actually remove that particular part.
Okay, I
Next
hope that
question, please.
answers your question, Sanjay. Uh, the next question we have is from Prabhat Kumar Swain. What are the state variables that you're looking for in the dynamic model in your study?
So, according to different, so far your dynamic model, the situation, environment is concerned, what type of model you are doing. So, in that way, you have to make the state variables. Just I'm showing the example, not my example, the example of my teacher, Professor Senrik Jorgensen. Suppose two people are working on SHIP model, SHIP, SHIP. So, one is actually how to minimize the friction with the water. So, in that case, the state variable will be different. So that's a SIP model. If another person is working, what will be the electrical wear is required to make the whole the electric line in the SIP. So they are set will be different in the same model, SIP model. So according to your problem, you have to choose which will be state of the system. So these are the state variables. Okay. So according to your problem, according to your model, you have to choose yourself who will be the or which will be the state variables. But depends upon you. Depends upon your problem. Depends upon your questions about the system. Next.
Okay. The next question is also from the same person. Can you tell some new technology that has been used in system ecology in recent times?
Recent times. So systems ecology, if you think about the systems ecology, the whole system, the only technology is the static modeling. But static modeling is developing in different aspect also. Every day, some things are added in the static modeling. But the basic backbone of the static modeling is not changed. Only one tool for the system psychology, whole system.
Okay. The next question is from Shorodip Dotto. He wants to know, is it possible to have more than one critical arc in a network?
Yes, it can be. It can be. But if you rate the critical arc, maybe, if you compare the height of 2 or 5 people, maybe 2 people having the same height, suppose 6 feet. But maybe if you make in millimeter, then 1 people height will be more. So in that case, two critical arc will not be the same value. Maybe they are critical. So now these are relative question. In which case it will be critical and up to which value it is not critical. So in that case it is very difficult. It's a relative question. But you can say from the critical means some arcs are more important than the other arcs. But which one arc is more important, other arc is little bit less important than the previous one, something like that. Anyway, any next question, please.
Okay, sir. Thank you, sir, for answering the questions. That's all the questions that we have today. And thank you so much for that wonderful talk. I would request Professor N.R. Banerjee to please give the vote of thanks.
Oh, it was such a fascinating lecture. Thank you so much.
Yeah, thank you, Anna.
And you explained it so nicely. So, from beginners to an advanced level, you have gone. And now many things are clear to me. Our conversation we would take offline, but for now, on behalf of the students of biology and also researchers in biology, I would like to ask that mathematics is always a sort of a bane for students of biology. So I would like you to share with us your journey from zoology through. I'm sure there are fascinating stories of how you chanced upon this field of study to where you are now.
So one thing I'm telling Ena, first of all, whenever I gone through the book of Colin Fox, in the preface it was written like that. I quote always. So without salt, egg without salt is tasteless. Okay. Sentence without grammar is meaningless, and science without mathematics is baseless. Okay. So what thing I'm telling you, whenever I started my ecological work, I felt that without mathematics, you cannot survive in ecological science. So in my PhD work, actually, I was not trained in a lab where the mathematical ecology or mathematics is actually applied. So I learned my own, the statistics and other things. But later, by my PhD, actually, I joined to the university, Jadavpur University, in the Department of Mathematics. And my mentor, two people I can acknowledge them very much initially. So they actually entered me in the realm of mathematics. One is Professor A.B. Rai, late Professor A.B. Rai, and Omiyo Bhushan Rai. Another person, still I am getting help from him. He is an incredible person for my career, for my job, for my everything. That is Professor Dilip Kumar Sinha, ex-Vice Chancellor, Vishwavarthi University, and ex-Provost Chancellor Calcutta University, and he was Raspberry Professor, Calcutta University. Incredible person. So they are incredible person. Actually I thank them for my initial development, and later I learned lot of things from very very good people. I am so lucky. I have visited so many lab, Milan Straskova's lab, even the Sven-Erik Jorgensen and Professor Patton, and even Professor Ulanovich. I learned a lot of things from him also, although I was not his lab. Okay, but I am thinking, one day I can just utilize, I can share. So on first day, whenever I went to Jadavpur University in the Department of Mathematics, Professor Abida, I actually bring me to the library of Jadavpur University, Mathematics Department Library, and he told the then librarian Keshavda. So Keshav just take the journal, that is the journal is ecological modeling. So he gave me 10 or 15 copies, volume of ecological modeling, and Professor Rai told me just to go through this paper, and you will be very lucky if you can publish any paper in this journal. And this is a God grace. I am now associate of this journal. So this is the journey I'm telling you. So I'm thankful to God also, because I met so much good people in my life, good philosopher, Professor Patton, Professor Jorgensen, Professor Straskova, Professor Eugene Odom. Thank you. Thank you.
As Professor Patton would say, that there is an innate connectedness of all systems, and that you have come across these people that much with so much humility. That means that you yourself are extraordinary human being and a scientist. So that I know you personally, but from what I heard today and the knowledge at the end of all the lectures, all the lectures that I have listened to, that is being
Your sound is not
the
coming properly.
description of that connectedness and the honest heart from you. So, with that, we end tonight's lecture.
Thank you.
So I think Ma'am is facing some technical difficulties.
Can you see or Professor Rai probably has some problem at his end?
Ma'am, can you hear us?
Yes, I can see and hear you, but where is Professor Rai?
Your
I think he got disconnected somewhere in the middle.
yeah, your speech is not very clear actually.
Okay, never
I
mind.
think it's because of
Never
the
mind.
connectivity.
You please carry on. Okay, that's fine. That's fine. You please carry on. Yeah.
As you all know, this is the last time we're meeting before a winter break, and we'll not be here with you all viewers for the next week. So during the holidays, please check out our website ppbiology.wordpress.com. It's a new website. We'll be posting lots of new content in the coming days. Please check out our website. Please check out our Twitter account, our Facebook account, our Instagram accounts, and subscribe to our YouTube channel. Just like you're watching us live, we also have a number of videos, a number of webinars which are archived, our previous talks. Please check them out if you have time, and please write to us on social media. Tell us what you think about us and the work we're doing. We also have a conference coming up in March of 2021. It's called the a half of it. The poster is out there on our website. We are very excited. We are happy to share the excitement with you. And enjoy the holidays, stay safe, and take care. Thank you everyone for being here with us today, and thank you, ma'am.
Yeah. Thank you, Rupkatha. Thank you, Shreya. And we wish all our viewers Happy New Year in advance. And we hope all our trials and tribulations of 2020 shall be over soon. So, with that, we end tonight's show. Thank you.
I just have one more thing to mention here. A lot of our viewers have been mentioning that there's a problem with the feedback link. Yes, we have figured that out, and we'll be posting the new link on the website in a few hours. Please bear with us, and please check our website in a few hours. So thank you everyone, and bye bye. Have a great holiday season.